Okay, so we've been to China, we've been to Japan, we're going to return to Europe now. Next, Professor Henry Markham from the Blue and Human Brain Projects.
So the Blue Brain Project is a Swiss-funded project, about 20 million per year, and about 7 countries, and is one of the precursors of the Human Brain Project. So I'll first tell you about that. The goal is a digital reconstruction and simulation of the brain. The fundamental scientific reason we believe this is possible is simply because the brain is an exquisitely organized system. There's over 60 years of mathematics and theory that shows that if the system is highly ordered, you can derive most of the structure by looking for the interdependencies between the parameters. So what we do is we hunt for the interdependencies, we build them into algorithms, and we create a hypothesis. The hypothesis is a digital reconstruction. Now, ironically, the easiest place to start is the most evolved part of the brain, which is the neocortex, because the neocortex is probably the most exquisitely organized structure. So easier than a snail or a worm. It is tractable because you can access the order. So as a proof of concept, we did a first reconstruction of a piece of the brain, and this was published last year. And this digital reconstruction is consistent with hundreds of experimental findings, not only at the light microscopic level, but also at the electron microscopic level. So this provides us with a hypothesis to test our understanding. It shows what we understand because that matches the data that has been obtained before. And we advance our knowledge when we can disprove this model. This is the fundamental principle of advancing knowledge in science that was established decades ago. It's not only structurally consistent with the data, and we can see where we can improve it to understand new aspects of the design, but it's also functionally consistent. So if we place this virtual tissue under a microscope in a chemical environment that is more equivalent to sleep, we see sleep-like states, and it mimics and reproduces experimental data that we see in the laboratory. If we now move it into an environment which is more like the wake condition, we then start to see, which it's not running, the movie, unfortunately, we start to see the kind of asynchronous and spontaneous activity that you see in the wake states. And without touching a single parameter, without designing the model specifically to reproduce experimental data, this virtual piece of tissue behaves and reproduces a range of very intricate experiments that have been done in the laboratory. Now, not only did we discover these two features, that if you build it and you follow the biology, that it will behave like a brain, in principle, but we also discovered many new things, that there is a spectrum of states from the sleep state to the awake state. And then what we discovered is that there's different coding strategies. The brain uses different strategies to code information along the spectrum. In other words, it's not whether you can solve a problem, it's whether you can change the state of your brain to that point in the spectrum where you can solve the problem. Now, fortunately, what we've also discovered is how to change that state. Which neurons to fire, which synapses to fire, how to change the chemical environment in order to change that state so that you can solve, in principle, any problem. And we're starting to understand that at a much deeper level. We're also extending this further to add in the blood vessels, the glia, the non-neuronal cells, simulating metabolism of the brain so we can understand the energetics of the brain. And we're taking that also on a larger scale to look at reconstructing, for example, the whole neocortex, which is already technically feasible. We still are in the process of modifying the differences in the different regions of the neocortex. This is a very early simulation of the somatosensory cortex, and we're seeing similar emergent activity principles as we see at the micro level, at this mesoscopic level. Then we did not build a model. We're developing the software to build any model. So we use the software now to, in principle, build any other brain region. And in the Human Brain Project, we're building many other regions in collaboration with many other scientists. So here, for example, we can use the same software and we run the same workflows to reconstruct the hippocampus. We're doing the cerebellum, the basal ganglia, and many other brain regions. So this is only going to go exponentially faster. This is an example of a collaboration we have with Allen Institute, where we essentially digest all the data they can generate. So we're very happy if the whole world is generating as much data as possible, because the software can ingest it, constrain it, see if it fits, and we actually discover a lot of errors in data because in many cases they don't fit, they don't make sense. We can go back to the experiments and we can check them. And so it's providing a powerful tool to actually test and validate what we do know about the brain. Here we used many different experiments from the Allen Institute, but also from many other laboratories around the world. This is just, again, a very early draft of the simulation of a whole mouse brain. And again, we're seeing very similar types of macroscopic behavior as we see at the mesoscopic and at the microscopic level. We are disseminating all the, well, first of all, we also have to plug that into the body. So we build virtual robots, virtual little mouse. And we can then begin to do virtual or digital behavior. In this case, we do whisker stimulation to see how the brain is going to process that information. And in the Human Brain Project, which is fundamentally funded by the European Commission, about 500 million over 10 years, and about 500 million from the partners, we are developing the infrastructure, the IT infrastructure, to be able to share all of this information and to be able to collaborate on an unprecedented scale. So, for example, we've built a collaboratory environment where 2, 10, 100, 1,000 scientists can sign up and say that they want to build the hippocampus, or they want to build a neuron or a synapse. And they can work together, distributed all over the world, to use all the same data, the tools, the software. It's available. It's even today. You can register. You can see the software. You can run the codes. You can test them. You can change them. You can say you've got a better solution. And in this way, we hope we're providing the very first step for international collaboration. It's a big team. There are many, many involved. These are the key scientists just involved in the Blue Brain Project's first delivery of its milestone. But the Human Brain Project is much larger. It's about 500 scientists. And we are very excited that there's so many other initiatives that have started so that we can ultimately lead towards a global initiative of brain endeavors. Thank you.